And sorry. And I'm very happy today to introduce you Chris George. I think that some of you have met Chris in our lab because he made 1 year placement from the University of Manchester, where he got his degree, first class degree, I would say, in physiology. And during that time, he moved to L'Aquila to work with us, especially on extracellular vesicles in breast cancer bone metastasis. So after he got the undergraduate degree, he got also a PhD at the University of Sheffield with Dr. Penelope Otwell. That is one of the most important groups in bone cancers in, I think, worldwide. So we've been very lucky, Chris. And in their lab, he's still investigating the effect of bisphosphonates on the immune response to breast cancer. And for his results, he got the ECTS Basic Translational Poster Focus Nestle Award in 2021 and the Physiological Society Academy Prize in 2018. So I'm very pleased to give the floor to George, to Chris, sorry, who will talk about defining the differential effects of Zoledronic acid under pre- and post-menopausal concentrations of estradiol on anti-cancer immunity. Chris?
Thank you, Anna, for that great introduction. I'm very happy to be presenting to all of you today after sitting through so many of these TMS presentations myself when I was there. As Anna introduced my project, I'll be looking at the effects of zoledronic acid under different menopausal concentrations on anti-cancer immunity. A little bit of background to breast cancer. It's still a big problem in the world, with over 12,000 people dying each year of breast cancer. In the UK, at least, it costs the economy a huge £1.6 billion per year. And although screening and treatments have hugely reduced the mortality of primary breast cancer metastasis still remains a big problem and is the leading cause of death, and it has few effective treatments. Now, breast cancer, as I said, can metastasize, and it has a number of sites where it preferentially does so. It can metastasize to the lung, to the bone, to the brain, and to the liver. And for my project, I focus mainly on metastasis to the lung and to the bone. Now, when breast cancer metastasizes to the bone, cells can arrive in the bone and can either outgrow or be maintained in a state of dormancy, either by the immune system or by the bone resident cells. But after some time, these cells can reawaken and they can grow. These grow into overt tumors quickly. These tumors release growth factors that act on osteoblasts and osteoclasts within the bone. This leads to bone degradation, which further releases growth factors that were in the bone, and these act back on the tumor cells themselves, causing those to outgrow again, which causes the bone to degrade, and it causes this vicious cycle of bone degradation and tumor growth. So what we try to do is break this vicious cycle to try to prevent the bone loss and prevent tumor growth. And one way we do that is using bisphosphonate agents. One bisphosphonate, which is the one that I use and is the most potent bisphosphonate drug, is zoledronic acid. This is a third generation nitrogen-containing bisphosphonate, which rapidly binds to the bone matrix after administration, and it targets hydroxyapatite within the bone. What it does is it prevents bone loss, and it does it by inhibiting osteoclast activity. However, zoledronic acid can also have direct cytotoxic effects on tumor cells. Because of this direct cytotoxic effect, there have been studies which have looked at trying to prevent bone metastasis occurring. So instead of using the drug when overt metastasis has happened, it's used as a preventative method. One of these studies was the Azure trial. Now, in the Azure trial, breast cancer patients were treated with zoledronic acid alongside standard of care. to try to prevent bone metastasis. So this was after primary tumor resection. And what was found was that treatment with zoledronic acid reduced bone metastasis and soft tissue metastasis, but only in the post-menopausal cohort of women, whereas the pre-menopausal cohort of women actually saw an increase in soft tissue metastasis. A number of preclinical studies have looked into this and tried to create models, and they've also found that zoledronic acid alongside endocrine therapy in mice before metastasis could also reduce bone metastasis. This was done by Penelope Otwell, who is my supervisor in my group. So just to summarize that study again. So breast cancer patients had their primary tumor resected. They then received standard of care treatment. And what this means is either chemotherapy or endocrine treatment, depending on the subtype of breast cancer. The patients then received zoledronic acid treatment, and there were differences in results between post and premenopausal. Postmenopausal women had reduced bone metastasis, reduced soft tissue metastasis, and an improved disease-free survival. However, premenopausal women, although they did have reduced bone metastasis, they actually had increased soft tissue metastasis compared to untreated premenopausal women, and they had a reduced disease-free survival. So the question is, what is the mechanism for this? Well, what we hypothesize is that the immune system has a role to play in this effect. And this is because both estrogen and zoledronic acid have strong effects on the immune response. Estrogen can affect a number of immune cells in the tumor microenvironment. It can act on T regulatory cells to increase their immune suppression, increase their activation, and increase PD-1 expression. They can also act on cancer cells themselves to increase PD-L1 and increase their proliferation. Furthermore, estrogen, as I said, can act on T regulatory cells. It can increase their trafficking into the tumor microenvironment and therefore increase T cell and natural killer cell suppression by T cells. Estrogen can act on T lymphocytes by decreasing their early T cell development, leading to thymus atrophy. It can also increase granzyme B inhibitor expression. This would mean that T cells would be less effective at causing cytotoxic cell death. Estrogen can act on macrophages. It can increase polarization of macrophages towards a pro-tumor macrophage. And what this means is that the macrophages would support tumor growth instead of actively clearing tumors. It can also increase MMP9 expression, which is very important for macrophage and tumor cell migration and invasion. Estrogen has also been shown to act on natural killer cells, reducing natural killer cell cytotoxicity and reducing natural killer cell activity in premenopausal women. On the other hand, zoledronic acid can also act on the immune system, and it can often do opposite things to estrogen. It too can act on T regulatory cells, decreasing their activation, therefore decreasing the immune suppressive environment within the tumor cells. It can also act on cancer cells themselves to increase phosphoantigen expression, which would mean that immune cells are able to recognize tumor cells more effectively. Again, it can act on a lot more than just T regulatory cells. Zoledronic acid, it can act on gamma delta T cells, which is a T cell subset. It can increase their activation by monocytes, again, increase their recognition of tumor antigens, and it can increase tumor killing via increasing cytotoxic granule production. Zoledronic acid can act on macrophages in an opposite way to estrogen. It decreases their polarization towards pro-tumor polarization and can increase their polarization towards an anti-tumor phenotype. It can do the opposite with MMP9, and it can reduce MMP9 expression, which reduces the invasiveness of tumor cells. Zoledronic acid has also been shown to act on natural killer cells as well, increasing their IFN gamma production and increasing their peripheral expansion. However, more research needs to be done on the effect of zoledronic acid on the immune system in general, which hopefully my work will contribute to. Therefore, we hypothesize that premenopausal concentrations of estrogen can inhibit the anti-tumor effects of zoledronic acid, via a PD-L1 induced reduction in T cell activity. So my study aimed to model the effect of estrogen and Zol on the immune response to cancer. We wanted to identify how these effects alter tumor growth in the bone and the soft tissue. And we then wanted to establish if a combination therapy with an anti-PD-L1 antibody, avelumab, can increase the efficacy of zoledronic acid in the premenopausal cohort. So the first thing we did is we wanted to have a look to see if zoledronic acid, if estrogen could affect zoledronic acid's anti-tumor effects. So as we can see, if we treat tumor cells in vitro with zoledronic acid, it leads to cytotoxic cell death. And that estrogen has no direct effect on this cytotoxic cell death. This suggests that the effects that were observed in the clinic are due to an indirect mechanism of zoledronic acid and estrogen acting through the microenvironment. Following this, we had a look at tumor cell migration. We did a migration assay. This is when you grow cells in a 12-well plate. You put a scratch down the middle of them. We then treated them with zoledronic acid and estrogen at post, peri, and premenopausal concentrations. And then we then imaged the scratch and calculated how much the scratch had closed to signify migration. And again, what we found is that estrogen and zoledronic acid led to no really significant effects on the migration of the 41 cells, again, suggesting that it is a microenvironmental effect, which is causing the difference in zoledronic acid effects between pre and postmenopausal patients. After looking at the effects in vitro, we then created an in vivo model to try to recreate the effects of the Azure trial. So we took mice and we overoptimized them to remove their native estrogen production and treated them with gosrelin, which also further reduces estrogen production by these mice. We then replaced this estrogen in their drinking water to mimic post, peri, and premenopausal concentrations. These mice were then given cancer by intracardiac injection. This is so that the cells were circulating to try to mimic the point in the Azure trial where Zol treatment was given. Mice were then treated with zoledronic acid or saline as control, and they were culled on day 19, and organs were taken: femur, tibia, lungs, spleen, and serum for downstream analysis. This was performed on two models, the C57 and the BALB-C mice. First thing we had a look at is the effects of treatment on hormone concentrations and bone turnover. The first thing we did was have a look at serum estrogen concentrations in these models. As you can see, in humans, premenopausal concentrations are between 200 to 500, and we saw that we successfully modeled estrogen concentrations to menopausal levels in our model. Furthermore, when we had a look at trabecular bone volume, we can see that estrogen administration successfully models bone physiology, which is observed in the menopause, and that zoledronic acid did not restore bone volume to premenopausal levels, as expected, since it's not an anabolic agent. We then had a look at osteoblast and osteoclast activity, and we can see that zoledronic acid significantly decreases osteoclast activity and significantly decreases osteoblast activity, which is to be expected since zoledronic acid's main action is within the bone, reducing bone turnover by killing osteoclasts. After confirming that our model is successfully modeling menopausal states, we then had a look at how our treatments could affect tumor growth and the immune response within our models. So, for tumor growth, our cell lines were transfected with the luciferase gene plasmid. They were stably transfected into cancer cell lines. This means that when we injected the mice with luciferin, which is the substrate to the enzyme transfected, it would produce visible light, which could be imaged using the IRIS system and could give us a quantification of tumor growth within our mice. What we found within the bone is that zoledronic acid could reduce bone metastasis, but only under postmenopausal concentrations. However, our effects within the bone between the two models differed slightly depending on the mouse strain. And this is a theme that continued throughout the project. Each mouse strain responded slightly differently to our treatments. However, our interesting results come from the soft tissue metastasis. And if you remember from the Azure trial, it is within the soft tissue that the main differences in treatment outcome is between pre and post-menopausal patients. In our BALB-C model, we can see that zoledronic acid can decrease soft tissue metastasis, but can only do so under post-menopausal estrogen concentrations. Whereas in our C57 model, we can see that zoledronic acid treatment under pre-menopausal estrogen concentrations significantly increased soft tissue metastasis. So taken together, these two results mimic the Azure trial in that zoledronic acid only having action under post-menopausal patients, and in our C57 model, treatment under pre-menopausal concentrations increasing soft tissue metastasis. We next had a look at the immune response, and we did this by flow cytometry. So what we did is we took lungs and bone marrow, we froze them down after taking them, and then we dissociated the tissues, trying to keep our cells alive. What we then did is we stained our tissues with antibodies and ran them by flow cytometry. So our antibodies have fluorophores on them so that we can identify different cell types by flow cytometry, and we can gate around them so that we can pull out specific immune cell subsets from within the tumor microenvironment. The first thing we did was we had a look at immune cell populations within the bone. What we found is that estradiol treatment significantly reduces the anti-tumor B cell populations in bone, showing that estrogen can have this pro-tumor anti-immune cell effect on B cells within the bone. We then had a look at natural killer cells, and again, we can see slightly different results between our two models, but we're seeing a trend towards decrease in natural killer cells following treatment with estrogen. So this is post, peri, and premenopausal estrogen, and then the same, but treated with zoledronic acid. Interestingly, when we had a look at M1 macrophages, we could see that zoledronic acid could significantly increase M1 macrophage polarization. This is an anti-tumor macrophage. However, it could only do so in the presence of low estradiol concentrations. Strangely, we saw a result that went against our hypothesis when we had a look at CD8 positive T cells. We could see that estrogen actually increased CD8 T cells within the bone following treatments with zoledronic acid and without treatment with zoledronic acid. However, T cells in the bone are at a very, there's very few T cells within the bone because it is an immune privileged site. And these results also do not take into consideration T cell activity. So although this is sort of goes against the hypothesis, further work needs to be done looking into this to see if these T cells are actually active here. We then had a look at immune cell populations within the lung. What we can see here is that treatment with zoledronic acid under post-menopausal concentrations of estrogen increase CD4 T cells, increase B cells, and can increase CD8 positive T cells. However, zoledronic acid can only do this under post-menopausal estrogen concentrations, whereas under pre-menopausal estrogen concentrations, there's no significant effect on T cells or B cell populations. This fits our hypothesis where treatment with Zol is only effective at post-menopausal estrogen concentrations, where at pre-menopausal, there's an increase in soft tissue metastasis. And what we're seeing here is in the soft tissue, zoledronic acid not being able to elicit an anti-tumor immune cell response. What we looked at next was PD-1 expression. So PD-L1 is an immune checkpoint inhibitor, which can be on tumor cells. The binding of PD-1, which is on immune cells, to PD-L1 on tumor cells, silences the immune cell response. Therefore, a decrease in expression of PD-1 on CD8 T cells would mean that they are less susceptible to silencing by the tumor cells. So what we can see is that zoledronic acid treatment reduces the expression of PD-1 on T cells which would suggest they could be more active. This is in the bone. Whereas in the lung, what we see is that estrogen significantly increases PD-1 expression on CD4 and CD8 T cells. Therefore, what we did is we wanted to see if combining treatment with zoledronic acid with an anti-PD-L1 antibody can increase their efficacy within the premenopausal cohort. So here it is. Does combination therapy with zolnivelimab reduce metastasis? So what we did is we created another mouse model. We took the exact same mouse model we used before, but this time we treated them with avelumab as well. So we either treated mice with control, with avelumab by itself, with zoledronic acid by itself, or with a combination therapy of zoledronic acid and estrogen. As a reminder, estrogen has been shown to increase PD-L1 expression. This means that it can reduce T cell activity within the tumor microenvironment. On the other hand, zoledronic acid has been shown to increase T cell numbers within the tumor microenvironment. However, if PD-L1 expression is high, these T cells will not be able to have cytotoxic effects on the cancer cells. So if we use an anti-PD-L1 antibody, we can increase T cells' cytotoxic effects on the tumor cells and hopefully reduce soft tissue metastasis in that premenopausal cohort of women. So these results here show tumor growth in the lung and the soft tissue. And excitingly, what we can see is that combination therapy with avelumab and zoledronic acid was able to significantly reduce both soft tissue and bone metastasis. This is a very exciting result. It's the first time that anyone has shown that combination with avelumab and zoledronic acid can reduce breast cancer metastasis. These results have been shown by another group to also be effective in a primary tumor model, and that this combination treatment can also reduce primary tumor growth. So all in all, it's quite an exciting move forward in combination therapy, I think. We next wanted to have a look at why this is happening. So we did some immune cell analysis. Strangely, when we looked at immune cells within the lung, so these are lymphoid cells following treatment with combination therapy, single therapy, Zol and avelumab, we found no significant differences in immune cell numbers within the lungs. Therefore, we had a look at immune cell activity instead. So we did PCR arrays and we had a look at granzyme B expression. Now, granzyme B is a marker of T cell and natural killer cell activity. And we found that granzyme B was significantly increased within the lung following combination therapy with avelumab and zoledronic acid. Furthermore, NOS2 was significantly increased. This is suggesting that although there's no increase in number of lymphocytes, there could be an increase in activity of lymphocytes within the lung. This is to be expected because if you remove the blockade of PD-L1, you might not necessarily have an increase in the number of T cells, but they will be able to have a cytotoxic action against tumor cells. We then had a look at immune cells within the bone, and really unexpectedly, what we found is that combination therapy with avelumab and zoledronic acid seemed to actually significantly decrease numbers of neutrophils, monocytes, natural killer cells. This really went against what we were expecting. So we wanted to try to understand further what this combination therapy was doing within the bone. Therefore, we looked at markers of bone turnover and we looked at trabecular bone volume. So just as a thought as to why this is happening, we can see that avelumab and zoledronic acid combination decreases immune cells within the bone, but combination therapy also reduces metastasis within the bone. So why is it doing this? Perhaps it's bone turnover. What we saw is that combination therapy with avelumab and zoledronic acid was able to significantly increase trabecular bone volume. Furthermore, it did this more than single treatment zoledronic acid or single treatment avelumab by itself. When we had a look at markers of bone turnover, TRAP, which is a marker of osteoclast activity, was found to be significantly decreased by avelumab and zoledronic acid. However, this was to a similar level to zoledronic acid treatment by itself. On the other hand, osteoblast activity, which is significantly decreased by zoledronic acid by itself, was found to be slightly increased following combination therapy. So what this suggests is that combining zoledronic acid and estrogen can prevent bone loss more effectively than zoledronic acid by itself. So what this suggests is that within the lung, combination therapy with avelumab and zoledronic acid are acting on T cells to increase their activity and tumor cell targeting, whereas the mechanism within the bone focuses more on the bone turnover markers, making the bone a less hospitable environment for breast cancer metastasis. In conclusion, we can see that inhibiting PD-L1 with avelumab can stimulate the anti-tumor effect of zoledronic acid. And what this could do is open up the efficacy of zoledronic acid to the pre-menopausal cohort of women where it was previously ineffective. So zoledronic acid and avelumab seems pretty good. For our future work, what we want to do is try to confirm if the effects of zoledronic acid on PD-L1 in the tumors. So we want to have a look at the effect of zoledronic acid on PD-L1 expression within the tumors under pre- and post-menopausal estrogen concentrations. We're also going to investigate the effects of zoledronic acid and estrogen on T cells activity and have a look at T cell subsets. So we've looked at numbers of T cells. We want to now look at further activity and more subsets. We want to establish how Zol affects tumor growth under pre- and post-menopausal conditions alongside standard of care. This is sort of the next step to taking this into the clinic, is having a look at how it interacts with normal standard of care treatments. And then finally, we also want to confirm these results using different models of breast cancer, either using a spontaneous mouse model or using a primary tumor model instead. I'd like to thank you all for listening, and I'd like to thank my lab, particularly my supervisor, Penelope Otwell, and everyone who's helped me out with my project so far. I will happily take questions from you all. Please be kind.
Thank you, Chris. Very, very clear. So I think that was inspiring. So we have people that wants to ask question. I would like just to ask Michela, since I know that you need to go.
Yes.
Soon. So if you want to ask,
yes.
Sorry, go ahead.
Yes. Thank you.
Switch on your camera, maybe.
Yes, sorry. Hello. It's very, very, very nice talk and very nice work. I'm very impressed. I would like to ask a technical question, since I'm going to use flow cytometry too very soon, I hope. And I would like to ask you, for the flow cytometry using tissues, as you told, no? You start from tissues. I would like to ask, did you use FICO stratification or you put a complete tissue? Because I think from, FICO from mouse tissues should be very hard to have a lot of cells. I don't know if you got my point.
FICO, sorry, I'm not sure quite what you mean.
Sorry, stratification to, to select only PBMCs, the cells for the immune system. Or you put...
So we used whole tissues containing tumors because we didn't want to necessarily just look at the percentage of cells that were, peripheral blood mononucleocytes. What we wanted to do is see how many immune cells had actually entered the tumors themselves. So because we were using the whole tissues and we're using lots of markers, what we could do is we could identify specific immune cell subsets by using a number of markers, and then we can do our statistics saying, okay, so this is the number of T cells we have. How many of these T cells are positive for PD-1? Or we can say this is all of the general immune cells by gating for sort of CD45 or CD11B, and then express our statistics as how many of the general immune cells within this tissue are T cells, or how many of these are B cells. So we're using the whole tissue so that we're keeping lots of viable cells, and then you're working out which cells with your backgating later.
Thank you. And also one more question, if I can. Is the effect of estrogens also act on, let's say, triple negative or breast cancer cells or cells that have no receptors?
Yes, so we've actually used triple negative cells for all of this study. And the reason we did that is because we hypothesized the effects of estrogen and zoledronic acid are not directly on the T cells, but they're acting on the microenvironment instead. Because, you know, we've seen that treatment with PD-L1 antibodies often work better on triple negative cells, and that the immune response is often more effective on triple negative cells because they express more surface antigens. So yes, we think that this could work for both triple negative and estrogen receptor positive cancers.
Okay, thank you.
No worries, thanks for your questions.
Okay, thank you. So Annabelle?
Hi, Chris. Thanks for a really great talk. It's really lovely data and presented so clearly as well. So thank you very much. So correct me if I've misunderstood, because I was just sort of pulling data together from two of your slides. If I recall, it was the Black 6 model that represented more accurately the soft tissue metastasis issue in the pre-menopausal samples. Is that correct? Before I
is this for the immune response, or is this for the?
So from the trial itself, the Azure trial.
So the Azure trial, yes.
You were representing it in the 2 mouse models, and if it was the graphs that you had on the right hand side of the slide, which if I recall is the Black 6 model, that was showing the increased soft tissue metastasis in the pre-menopausal.
So what we had was the Black 6 showed that treatment with zoledronic acids under pre-menopausal increased soft tissue metastasis, but our other models showed that zoledronic acid was able to decrease soft tissue metastasis only under post-menopausal. So they both showed aspects of the Azure trial.
Perfect. Okay, so in that case, my question therefore is, when you've done the in vivo model of the combination therapy, and you showed us with the BALB-C mouse that it was having this really nice effect and still reducing that soft tissue metastases, have you looked at that combination treatment also in the Black 6 to see if it's able to reverse the sort of detrimental soft tissue metastasis effect there in that model as well?
We have not done that yet, but it is on the cards to do. So far, we've only been able to look at the combination therapy in the BALB-C model, but our future work, which we've got grants in for, to try to expand this into lots more models. And of course, we saw lots of, mouse strain specific differences in treatment outcomes. So I think it's really important to do those studies, But unfortunately, we haven't done it.
Yeah, no problem. That's fine. It'll be really nice to see that data, because that's where sort of the issue is with the Zol treatment. So it'll be really nice to see, hopefully, if that's rescued. Brill, thank you so much.
No worries.
Thank you, Anna. And Hannah Ford.
Yes, hi. I'd also like to agree with everyone that it was a really nice presentation and really interesting. But my question's more on the background. So do you know any reason why estrogen has these kind of immune response reducing effects, like from an evolutionary point of view, or how does it benefit premenopausal women, or do you not? Is this kind of unknown?
Let me have a think. I would have written about this in my introduction.
It's a tough question.
I didn't know if you knew any of the reasons why this is.
I think it might come down to to produce excess inflammation in premenopausal women, but I don't think I can give you a particularly in-depth answer about it.
Okay.
This is absolutely true. So when the estrogen level drop, then you have a kind of increase of inflammation, and this is why some authors think that osteoporosis is an inflammatory disease. So you have an upregulation of many pro-inflammatory cytokines. So it's absolutely possible that there is a connection.
I think research into osteoporosis and more immune-related effects is very interesting.
Yes, more and more there is a connection between osteoporosis and immune response, and also for other diseases. Yeah. Are there any other questions? I see some. thing in the chat. Oh no, it's Annabelle. That's a great question. Katie?
Hi, perhaps a bit of a naive technical question from me. I was just wondering, what is the benefit and why do we use different types of mice strains in these investigations?
Yes, so as you can see from my data, each mouse strain will respond slightly differently. So, and also they have dual, and that had E0771 cells, but C57 mice have, I think they have a preference towards a Th1 immune cell response. And if you look at BALB/C models, they can have a different preference for their immune response, I think often eliciting sort of Th2 responses. So by using different mouse models, you're sort of representing more the heterogeneity, which is seen in the patient population. Whereas if you're just one tumor cell type, one mouse model, you're sort of railroading yourself to this is only happening here. So it's a bit less translatable.
That makes sense. Thank you.
No worries.
I see Annabelle again, or is your previous? Okay.
Yeah, it's just sort of a follow-up question / comment from what Katie's just said there. I wondered if you've considered using what's considered sort of a humanized immune mouse model, where you could put a human peripheral immune system into a mouse model, whether that would give you any more insight using sort of specific patient received bloods to form that mouse model to give you a better idea of patient heterogeneity.
Yeah, I think that would be a really interesting model to use. I don't think we've really thought about using it, but there would definitely be benefits from that. For instance, one of the cell types that zoledronic acid affects the most is the gamma delta T cell subset. I briefly spoke about it in the introduction. The issue is that mouse immune systems don't really have any sort of comparable gamma delta T cells to humans.
Okay.
Don't see that response at all, and it's quite a major population and affect a cell for zoledronic acid. So yeah, I think if a system like that were able to robustly mimic the immune system in mice, then it would be really useful to use.
Yeah, that could be great. If you do consider looking into it, the model, which is a mostly sort of T cell grafting model, would be literally just the injection of PBMCs into the mice as opposed to the CD34 model. So if you're considering it, just to sort of bear that in mind, that would give you more of a T cell grafting than a B cell or other immune response graft, but it might be nice to look at.
Yeah, that would be good. Thank you.
I see a collaboration coming up between Chris and Anna. Michaela, you have another question? Still here?
Yeah, because they are late. So
okay, good.
I'm lucky. So yeah, I would like to link with Hannibal because it could be useful maybe before to treat in vitro, maybe PBMCs from human, because you can easily take it from circulating blood. Maybe you can co-culture with, I don't know, tumor cells or osteoblasts or some, you can make an in vitro, let's say, simulating test before. Maybe it could be nice to check the effect. I don't know if.
I totally agree with you. I think
easier.
Yeah, we discussed these experiments, doing more in vitro immune cell data and taking PBMCs would be really good. Another thing we were going to do was to isolate from our mice spleens and from tumors to isolate immune cells and then try and do co-culture experiments in vitro. We started doing lots of setup for this, but then halfway through we sort of realized that this project alone could be a whole PhD project, and there might not be time to fit it into this one PhD, but it's definitely work that we want to do. And I think
I'm thinking just human PBMCs because maybe, for sure, you know that mouse immune system is a little bit different.
Yeah.
Also the spleen, no? So I don't know if maybe it could be more useful to use human PBMCs instead of mice, but
I agree.
You know, do both is better than.
Yeah, but then we could also look at human cancer cell lines, and it would make it slightly more translatable, I think. So yeah.
You know, Chris, this very much will depend on if you will stay in the field or if you will change field. Because if you will stay, I think that you get a number of ideas, and so I think that this is absolutely important. Anna, do you have another question?
No, I just thought
Okay.
I'd leave my camera on rather than going
Okay.
on and off.
Okay, we have Flavia. Go ahead, Flavia.
Yeah, hi, Chris. A very good talk. Thank you, because it was extremely insightful. Hi. Just keeping an eye on the fact that all our kind of research and development work then has to go straight forward to the clinical trial, hopefully. My question for you would be, given the fact that you analyzed very well the zoledronic acid effect with respect to the avelumab-1, would you suggest as a kind of general practice to understand via clinical trials the effect of zoledronic acid as a kind of boosting agent for improving the performance and the clinical outcome of other anti-tumor drugs in general terms?
Yeah, I think that zoledronic acid could, because it does seem to have these immune stimulatory effects, it could definitely be used in conjunction with other drugs. It's a well characterized drug that's already used for a lot of diseases. So being able to use zoledronic acid to improve other treatments would be quite an easy thing to do, I think, to get into the clinic once you've done those base studies. So I think it has a lot of potential. I think it's also been overlooked a bit in the past because it is a bone targeted agent. So the sort of general consensus is that zoledronic acid binds to the bone and is only active there. But what we're seeing is that it seems to be having more of a systemic effect. So I think it is an exciting new sort of a new time for bisphosphonates to come back after they've sort of been written off to other drugs. But yeah, I think combination therapies would be useful. So there's lots of other checkpoint inhibitors it could be used for cancer. And it can work the other way around as well. I mean, if you look at avelumab itself, it's actually pretty ineffective in most solid tumors. In breast cancer alone, avelumab only really works for triple negative breast cancer or women who have high PD-L1 levels in their tumor. So combining it with zoledronic acid could allow it to be used with, a much wider subset of women for these cancers.
I see. Okay, thank you.
Still is the best therapy for bone metastasis yet, although it's not that strong, but I think that we really need more information. And you're right, Chris. I mean, the problem is that there is this great affinity for bisphosphonates with the hydroxyapatite, but this is not the whole story, because they also have some direct interaction with cells. So for sure, yeah. I have a question, a suggestion. I don't know. I mean, it's something that there is one cell that is missing in your investigation that are the osteoclasts. You mentioned, of course, that they are target of bisphosphonate, but I want to know if you have thought through the other function of osteoclasts as the immunomodulator cells. This is absolutely very in fashion now, and it's clear that the osteoclasts are heterogeneous, and some of them are really suppressor cells. And so it could be good to manipulate these cells, for instance, to improve the immune response against the tumor. I don't know if you have data on that, or if you think that is something that maybe in the future can be done.
Yes, well, I think that this also might be one of the main actions of zoledronic acid alongside avelumab within the bone, and why it is so effective there for both the pre and the post-menopausal conditions, because removing that immune suppression of the osteoclast within the bone alongside avelumab, which is stimulating the, removing the sort of the blocking of the immune system on the tumor cells, is sort of a double-edged hit in the bone. And I think that it would definitely be something worth looking at. Yeah,
also because these osteoclasts have a lower bone resorption activity, so they're mostly oriented towards the immune response rather than bone resorption. So I think that is something that could be investigated, and maybe if you have in your archive material the possibility to look the status of disaster. Another important and interesting observation is that the heterogeneity of the osteoclasts very much depend on the source, the cell source from which they originate. If they are dendritic cells, if they are classical monocyte, and so on. So something that maybe in this context, I think could be important, because you are modulating the osteoclast certainly by zoledronate, but perhaps also through the immune therapy.
Yes.
So something that probably has a meaning.
Well, we did see a small effect on osteoclast following just treatment with avelumab. There seemed to be a decrease in osteoblast activity following just avelumab. So it's having some effect on the osteoclast, probably through the immune-mediated effects. Yeah.
Sure. Yes. Interesting. Any other question, comments, ideas for Chris?
I have a non-scientific question if we have a minute.
Go ahead. Sure,
So,
yes, we have time.
I suppose for all of us who are following a similar academic route to you, to ask what are your next steps and what's your sort of hopes career progression wise, as one of the first of the L'Aquila alum to be moving forward from your PhD?
Yes. Well, right now, I mean, I've just submitted my thesis and waiting for my viva. So I'm actively looking for postdocs right now. I want to stay in the sort of the fields I'm in right now. So really staying in sort of cancer metastasis, the immune response, immunotherapies, or conversely, staying in bone cancer. So I've got quite a few options and roads to go down. So personally, what I want to do is do a sort of 2 to 3 year postdoc project. And I want to move abroad for that time to try to get as much experience as I can in different countries, different labs. Following that, I'm not too sure what to do. I know that that a life as a postdoc can be quite unstable for the first sort of 10 years of your time. So depending on how the first postdoc goes, I'd have to see whether I'd want to make a transition to industry, whether I'd want to stay in academia. I'm not too sure for now, but definitely a postdoc next to try to get a few more publications under my belt. To get to a country where it doesn't rain every day, that would be nice.
And Chris, you know that you have a support from me. So I've been the reference person for many of you, and I'm very proud that all those who were looking for a PhD position, they got it, and now it's the postdoc turn. So I'm very, very happy. And maybe, Anna, we can announce that in June we will have a webinar on life in industry, I would say, because we would like to give some information to the audience about the many different possibilities you have in science. It's not only being in the lab, not only being in academia, not only being a professor. There are many other top level possibilities. So we are working in this direction, and in June you will have a webinar on this aspect, especially linked to industry. So any other comment? Michaela again here. So Michaela.
Yeah, they didn't start yet.
No,
so.
because there is some overlapping, and we were a little bit worried because how we could handle that. Instead, they are always late, so we can make our presentation without any problem. So go ahead.
So one other maybe possibility is to check easily, let's say, the level of release factor from immune cells, like in the serum, no? So when you use this drug, you can check also by ELISA or something like that. If there is some changing in interleukins or something like that, could suggest you some, I don't know, some new ideas, some new changes.
Yeah, I think that's a great idea. Some things that we're putting together now for the paper, are we going to be looking at? Firstly, we're looking for Western blot for more PD-L1 expression, for granzyme B expression. And we have been suggested to do a few more ELISAs as well.
Okay.
We've also designed a new flow cytometry panel which focuses more on subsets of T
Yeah,
cells.
subpopulations, yeah.
Yeah.
Really targeted, but yeah, no, thank you.
Yeah, also you could take advantage of the public data, large-scale data that are available in terms of transcriptome, proteome, and so on, and maybe make correlation between the various therapies, ER status, treatment with Zoledronic. So this is also something absolutely possible and not difficult because they are available free on the platforms. And they are also very popular, I must say, in papers. So you don't publish if you don't have one of these analysis. Takes just a few minutes, I think. Okay, questions, comments? Anything else you want to know? I hope that you enjoyed this really clear, very well presented webinar, and I'm very happy to see your progress, Chris. In 3 years, you really did a lot of work, and this is absolutely nice. So if there are no further questions, I think that I will thank everybody for attending this webinar. And Chris, if you want to be in touch with us, as ever, every Thursday, 1:30, we have a webinar, and I don't know if you are still in the mailing list, but you can do that and receive the information. Anna, do you want to say anything?
No, just good luck for your Viva. I hope it all goes really well, and good luck for the next steps. It'd be great to hear where you end up next.
Yeah.
Yes, thank you. Same to you. It's been great talking again and being on the side of TMS.
Yeah, I have actually another question. Are you attending the ECTS Congress this year?
No, I'm not this year.
Oh, you're not. Okay.
But
Because that was a good opportunity to meet.
What about EACR Congress?
Oh, okay. Okay. Okay, so thank you very much. Good luck for everything, Chris. Thanks, Anna, for your great job as chair of the TMS. And thanks, everybody, for participating. And in 1 week from now, of course, we will have another event. Thank you. Bye-bye.
Thanks very much. Bye.
Cheers. Bye.
Bye-bye.
Mm-hmm.